This is a tutorial to demonstrate how to use the SRA Webin tool for the submission of metagenomic sequence data to the Sequence Read archive, complete with the Genomic Standards Consortium or GSC compliant metadata. For more information about the GSC, please visit their website at www.gensc.org. Before beginning a submission, you will require an account. Use the Create a Submission Account link on the EBI Metagenomic submission page. As every request for an account is manually audited, this may take a few days to receive your login details. Once you have them, click the SRA Webin link on the EBI Metagenomic submission page, and you should see this page. You may log in with your login details. To create a new submission, click New Submission. You will now need to select the appropriate option. In this case, we are going to make a complete submission, so we'll leave the default option. Before completing a submission, you will need to upload your sequence files. We suggest using the SRA file uploader by following this link. It's a Java application, and you will require at least Java 6 to be able to use it. You will need to log in again using the same login details as your SRA Webin account. Search for your directory in which your files are stored. And open that directory. It will show you all of the files that you have here. Select those that you wish to upload and click Upload. As these are mock files, they are very small and take less than a second to upload. If your files are very large, this may take some time, so you can simply minimize this box and return to the submission, SRA Webin submission tool. On this page, we will need to enter our center name, which is given at the time of creating your submission account, and select a release date for your data. The default is 2 months in advance. It can be set up to 2 years in advance. Click Next. If you are submitting information relating to an existing study that you have previously submitted, you may select it from this list, or to create a new study, click to New Study. You will be required to enter a short unique name. This is unique to you. It does not get you on a public website. It is for your reference. So in this case, I will use CowTest. We are demonstrating the submission of metagenomic data. We need to select type. All metagenomics data goes under the type of metagenomics. This includes shotgun metagenomics and amplicon metagenomics. We are then asked for a more descriptive title. This is the title that will be shown on the public web pages when your data becomes publicly visible. And you'll be asked to provide a full description. This can include either an abstract from a paper that you are writing or all of the information about the study. Once you have written your description, click Next. You will then be given a selection of checklists to choose. As we are submitting cow metagenome data, this is host associated, so I'll select that one. Note the upload spreadsheet option. This can be used by more advanced users who have previously created and correctly formatted the spreadsheet containing all of the sample metadata. This option will not be covered in this tutorial. On this page, you are required to personalize your template by selecting appropriate terms for which you wish to provide details from the panel on the left-hand side. Certain terms must be included for your data to receive the Genomic Standards Consortium Minimum Information Compliance stamp. These have already been selected for you and cannot be unselected. If you are unable to provide any of these GSC required data, please contact our curators by emailing datasubs@ebi.ac.uk for assistance. We now need to add some extra terms, so you can search through these to find the things of interest. So we're going to add those information, or if you don't want to scroll through these pages of which terms you wish to add, of which there are quite a few, and you have an idea of what you want to add, there is a filter option at the top here. So if we want to add a tax ID, you can type host in there and it will give us all of those that begin with the word host. Maybe we want to add the colour of the cow as well. If you are unable to find an appropriate heading, you can, if you wish, add your own attribute by clicking add once you've written it there. Once you've selected all the terms for which you can provide data, you can now input the common details for all of the samples in the template file on this side. So if all of your sample names begin with the word cow, for example cow 1, cow 2, cow 3, etc., that can be put in there. Similarly, all of my metagenomes are from rumen, so I could put in the search engine the term metagenome as it's a metagenome, or I should be able to find the bovine gut metagenome. Investigation type will either be metagenome for whole genome shotgun, or MyMark survey for a marker gene survey, such as 16S. My project name was cow test. My sequencing method for all of them was parasequencing. All the samples I made were collected on the same date. All of my host are cows, and the tax ID that I happen to know for that, because I've been looked at previously, is 9913. Each of my subjects has a different ID, so I will give an individual cow identifier in that space for the individual samples, and they're all different colours. The environmental package is host associated as selected on the previous page. The latitude and longitude is given in decimal degrees. Country is a list, so if you start typing a name of a country, it will give you the options. Once you have put in all of the details that are common to all samples, I would like to find the term diet and add that to my list, which has now been added here. Click next. You are now asked for how many samples you are submitting data for. It is just 2, so I will click add. There is now a list of all of your samples by the unique, so it includes the prefix which you gave on the template page, plus the sequential number. You can change the name if you have a more specific name, or if you wish to simply alter it by adding in there. Each sample will require a type. And a full description of the sample. For human consumption, so this is the human readable description that's given with each sample on the web pages once it's public. You're looking to get a tick in each of these terms down this side and to provide as much detail as you can. So this was cow 1684, and the host colour was black and white. Its diet was fresh grass. All of the details are complete. I must now click Update in order to make the changes which become validated here. I'll move to cow 2 and complete the details for cow 2. Must click Update. I now have a valid tick for both of my samples, and I next. Because these are 454 files, they must be in SFF format. I uploaded the SFF files at the beginning. You may now add the number of files that you have and select the appropriate names from the drop-down lists, the instrument model details from the drop-down lists, a library name should be added. It can be something appropriate. Or if you do not create a library, we have to make something up. Select where that was from, and then the file name as given to make the upload. And click Submit. Please confirm that you want to see your data. OK. This is the test server, because I was not willing to make up one to put onto the public servers. If this were the production server, you would now receive your accession numbers on page. So if I wanted to go to this production server, I would click this link and repeat the process on exactly the same way to actually submit my real data. Thank you for listening.